Morning, everyone. I'm Suzanne Paley. I'm one of the principal developers behind BioCyc and Pathway Tools. And so I'm here to tell you about some of the great informatic capabilities we have. So let me start by clarifying what I'm talking about here. What are BioCyc and Pathway Tools? The BioCyc database collection is our collection of over 20,000 organism databases. Most of them, but not all of them, are prokaryotes. The BioCyc website is where you access all this data. You can use it as an encyclopedia to look up information, or you can use some of our many tools for omics analysis, comparative analysis, and more. And then the Pathway Tools software is the software that we've developed to create, manage, and host all this data. It's what powers the BioCyc website, but we also make it available for you to download if you want to, say, build your own databases for your own genomes or run your analyses in-house. So we call each of our databases pathway genome databases, or PGDBs. You may hear me refer to that, because they combine a very rich set of data types, from genes and genomes to pathways and reactions, enzymes, transporters. And when I say pathways, I'm primarily talking about metabolic pathways here. The vast majority of our 20,000 databases are purely computationally derived, with no manual oversight. But we do have a growing number of curated databases. We're focusing on organisms where there is large community interest in literature, so important model organisms, important pathogens, etc. And I particularly want to draw attention to our flagship database for E. coli, which is BioCyc. We've been curating that database for over 30 years from the primary literature, and it's the preeminent E. coli reference database out there on the web or anywhere else. Most of our databases describe a single organism each, but I also want to call attention to BioCyc, which is our encyclopedia of metabolic pathways, reactions, and enzymes from all domains of life. BioCyc is painstakingly curated from the primary literature. Our curator, my colleague Ron Caspi, who also serves on the Enzyme Commission for EC numbers, generates these mini reviews for enzymes and pathways that people really like. These reviews. They're like mini textbook pages. Much as we might wish for all these LLMs out there to go and read the literature and create these lovely summaries, they're just not up to the level of an expert human biologist. So just so you know, you can trust what you read there, because Ron doesn't hallucinate. In addition to being a superb reference source, BioCyc also serves as the cornerstone of our database generation pipeline. So let me just tell you a little bit about that. We start from a fully annotated genome from RefSeq or somewhere else. So we're not actually doing any gene annotation here. We're using the gene annotation that expert annotators know what they're doing about. And then we build a database out of that, creating objects for all the genes, proteins, RNAs, et cetera, in the annotation. And then we use BioCyc as our source for metabolic reaction and pathway data, and we predict the reactions and pathways in that organism. We also predict the transport reactions. We search the genome to try and fill any holes in the pathways that we predict. We have algorithms that predict operons. Remember, we're talking about prokaryotes here, and protein complexes. We compute PFAM domains. We compute the orthologs between all our databases. We can also import data from other databases, such as UniProt or PSortDB. And that gives us what we're calling a tier 3 or uncurated database. And then for a few selected databases, we go on to add additional manual curation. So when a curator curates one of our databases, they're essentially doing two things. They're reviewing all our computational predictions, making any needed corrections, deleting the stuff that we over-predicted and maybe adding new things. And then they're adding new data from the literature. So this can involve both individual changes to particular proteins or other kinds of objects, but also uploading any large data sets that they find out there, such as for transcription start sites or small RNAs or whatever they can find. In addition to being a great general resource, or reference source, we also offer a huge selection of tools and other capabilities on our website. So lots of different kinds of searches, lots of different visualizations, omics analysis tools, comparative analysis, metabolic modeling, etc. And I certainly don't have time to go into all of them today, but I'm going to survey a few of them. So we like to say that BioCyc lets you do better science faster. So by synthesizing all this information and putting all our analysis tools in one place, we save you time. We give you a one-stop shop where you can work with all our stuff together. Our site is easy to use, and our various visualization tools let you create professional figures in minutes. Now for the bad news. EcoCyc is grant supported, so access is free for everyone. That's not the bad news. But if you recall on Friday when Fiona was talking about the various approaches that people have come up with when the grants are no longer supporting their ongoing operations, our solution to that is to require a subscription for all our other databases. We're not for profit, so all our subscription revenues go into our curation. They go to support the curation of databases other than EcoCyc. We offer institutional subscriptions, so check if your university has one, as well as individual and lab subscriptions. So if you like our site, bug your institutional library or your advisor. We also require our users to sign up for accounts on our website. And that is true even if you don't need a subscription, because if your organization already has one or if you're only accessing EcoCyc. Creating an account is free, and the good news is that with your new account, you get 1 month free access to all of BioCyc to try it all out. So I'm going to briefly survey some of our site capabilities and then pick just a few to go into more detail on. We offer a wide variety of tools for gene and protein informatics. So you can search genes, gene products, other genome sites by a whole variety of attributes. I don't know if you can see my cursor. There we go. Here are some images from our newly updated genome browser and our comparative genome browser. I just gave a talk on this yesterday, but if you missed that, you can look it up or you can catch me later and I'll tell you about it or show you it. So our new genome browsers, it offers rapid, precise zooming and navigation and some other nifty features. This is our circular genome viewer. And we also offer a variety of sequence-based capabilities. So blast search, sequence pattern search, and multiple sequence alignment, which is shown down here. This is an example of one of our gene pages. It's got links to sequence information and comparative operations, links to our genome browser, reactions and pathways. The summary contains, I don't know how well you can see this, but this is our regulation summary diagram that is automatically generated, but summarizes all the regulatory influences on a particular gene and its gene product. And then we have our textual summary here full of citations to the primary literature, links to other databases. This is an enzyme. So the reactions tab lists all information about its different reactions, including the pathways it's in, the activators, inhibitors, cofactors, kinetic information if we have it. If this were a transcription factor, we would have a Regulon tab instead. I'm not going to show you all the tabs, but here's the operons tab also, which shows you the operon organization and the position within the genome and the binding sites and transcription start sites, etc. We also offer a wide range of capabilities related to metabolism and pathway informatics. We have a whole slew of ways to search for pathways, reactions, and metabolites in various ways. This is an example of one of our pathway diagrams, which are computationally generated, but high quality and look like the kind of thing you might find in a textbook. You can customize the pathway diagrams, and you can create customized multi-pathway diagrams based on selected pathways. We call these pathway collages. That's a different tool. We automatically compute atom mappings for most of our metabolic reactions, and then we show them color-coded on our reaction pages. Here's an example of a color-coded reaction. You can also have an option to actually see atom numbering, so you can see where atoms in the reactants line up to the products. We create organism-specific full metabolic maps, and you'll see an example of that in a couple of slides. We also have a metabolic route search tool where you can enter a starting metabolite and an ending metabolite, and we'll try and search for likely paths between them. And then we also have the ability to build quantitative flux balance models. Here's an example pathway page. Notice the textual mini review here. And this is our pathway diagram, and you can easily change the level of detail to say show compound structures. And so we offer a few sort of canned detail levels, but you can also customize it. Although we don't computationally infer regulatory data, we can capture it if it is available. So transcription factor sites, that kind of thing, we can capture it. So our curated databases will often have this kind of data. This is an example of our metabolic summary diagram that I mentioned a couple slides ago. So this is for E. coli, and I've actually gone and highlighted in red here all enzymes and transporters that are regulated by the transcription factor FNR. So you can see that in a pathway context. We have a whole suite of tools for omics analysis visualization, and I'm actually going to go into some of these in a bit more detail. This is an example of our top-level view for our omics dashboard. Don't worry about it. You can't read it. I'm going to describe it a bit more shortly. We've recently added support for multi-omics data sets to this tool. We also offer a variety of ways to map omics data onto our pathway visualizations, whether that's on individual pathways or our customized collections of pathways, or on the entire metabolic network. We offer several different kinds of enrichment analysis, both through our omics dashboard and through our smart tables, which I'll also describe again in a few minutes. And we have some other things too. So this is an example of mapping transcriptomics data onto our metabolic map. So this particular data set is a time series data set with 5 time points. On the full map, you can only see one time point at a time, but you can actually play it as an animation, so you can see how things change over the course of your experiment. And then you can focus in on just a few reactions of interest and show these bar graphs for just those genes over the entire series. And this is a section of a similar diagram, zoomed in a bit so you can see the pathway labels, but this has multi-omics data, which this is a recent new feature for us. So here you have reaction edges colored according to the transcriptomics data, and then the metabolite nodes are colored according to the metabolomics data. And so you can see where the trends of both are aligned, like here, or perhaps where they might not be somewhere else. We can also use node size or edge thickness as additional display dimensions. So if you have additional kinds of data, you can include them, or if you want to include significance scores for thickness or whatever, you can do that. This is an individual pathway diagram, and we've overlaid our omics data with it. This is transcriptomics data here with plots. Bar graphs, again, this is a time series. So for each of the reactions, we've overlaid plots for each of the individual genes. And this is, we've computed a table of the most perturbed pathways from our input data set, and we're presenting them as a table with the omics values. I don't know if you can see them, but they're little colored boxes next to each gene name in our little pathway diagrams. So those are our displays, or some of our displays on actual pathway diagrams. I'm also going to talk about the omics dashboard, which I mentioned earlier, and which provides a different way to explore your omics data. So it also supports multiple types of omics data, including, as of our latest release, multi-omics data. The idea is to initially provide a broad-based overview of many high-level cellular systems, and then let the user successively drill down into subsystems of particular interest. So on the earlier slide, I sort of flashed you a picture of the top-level display, but you couldn't read it. So I'm going to take one panel as an example to show you just what we're doing with the data here. So this is the energy metabolism pathway panel. It's divided into subsystems for glycolysis, the TCA cycle, et cetera. This is time series data. So each of the colors correspond to different time points. Each of these small dots correspond to individual expression values for the different genes associated with the particular subsystem, in this case, fermentation. And then each line also has a large dot, which gives the average expression value for all the genes in the subsystem. So at one glance, you can see how the range, the distribution, and the average expression value is changing over the course of your experiment. It makes for a very information-rich display.
Average for
the mean. So here's that, or a portion of that top-level display again. Suppose I want to look deeper into amino acid biosynthesis. I click on the plot to get a panel that shows a similar kind of display, but now the subsystems are for biosynthesis of individual amino acids. If I want to drill down further to look at arginine biosynthesis, I click on that plot, and now I'm at the level of the individual genes involved in arginine biosynthesis. From here, I can look at the actual pathway diagram overlaid with my omics data, or I can look at the operon diagrams. And then with multi-omics data, which is again our new feature, we're just displaying the different data sets side by side in each subsystem. Here we happen to have 6 time points with metabolomics data and 2 gene expression time points. And again, each time point gets its own color. Now they're all displayed together. We have the option of using dual y-axes. So if the data aren't normalized, to use the same scale, or if one of them is absolute quantities and the other is fold changes, they all show together, but each with their own scale. The base level panel shows both the metabolomics data and the gene expression data together. So you can see both the metabolites and the pathways and how and the genes, again with the dual axes, so you can see how they're acting together. And then again, our pathway diagram has both the metabolomics data and the gene expression data there in the diagram for you, with again multiple scales. So switching gears now, I want to tell you about one more very powerful and popular feature of our website, and that's the ability to create and manipulate what we call smart tables. These let you define lists of objects of pretty much any type. You can explore their properties, transform them in various ways. You can filter and combine them. You can make them public or just share them with specific collaborators, or you can export them to a spreadsheet or some of our other tools. You can use them for omics analysis, either by directly running an enrichment analysis on a smart table or by exporting them to one of our other omics tools. And so here's an example of a smart table where we've started with a list of genes. We've added a column for the products. We've added another column for the pathways that the gene participates in. You can create your own smart tables either by uploading a file, or pretty much anytime you run a query on the BioCyc website where you get multiple results, our pages will give you the option to turn the results into a smart table. You can access smart tables that other users create if they either make them public or share them with you. And then we predefine a set of what we call special smart tables. These are things like all genes in the current organism, all pathways in the current organism, all enzymes, etc. And then a smart table contain both PGDB objects, but also other fields like numbers, like your omics data, perhaps. I mean, here's just another example of a metabolite smart table. These are metabolites. We're also showing the SMILE string and the compound structure. And this is just one more example of a gene smart table, this time with the sequences in there. You can do a lot more things with smart tables, but I don't have time to go into that. I do encourage you to check them out. They are very popular. So again, come check out the BioCyc website. Sign up for a free account. Try it out for a month. If you find it useful, encourage your institution or your advisor to subscribe, or install the Pathway Tool software locally and build PGDBs for your own organisms and use the same tools. And we have a whole suite of training videos on our site where you can learn more. We are pretty responsive to user requests for help or suggestions for features, that kind of thing. So do contact us if you have any issues. And I think that's out of time.